influenza. Any questions so far? Are you guys following me?
Yes.
Yes. All right. So 3 types of influenza viruses. We have A, B, and C. And as I said, there are many strains, and the strains are separated or categorized by antigenic differences in the glycoprotein spikes. Okay? Now, type A infects humans. It can infect other mammals like swine, and it can also infect birds. Type B and C only has only been isolated from humans. It only infects humans. So, when we study the influenza disease, there are 2 important questions that we ask ourselves. If an antibody to the NA and HA are protective, if we have antibodies against these 2 glycoproteins, why is it that we always get epidemics from the flu? Okay, why? The answer to that is antigenic drift. Okay, so let's see what happens here. During antigenic drift, what happens is that during the viral replication, mutations can occur on the neuraminidase or hemagglutinin spikes, leading to changes in these antigenic glycoproteins. Okay, this is what's called an antigenic drift because the changes are small. This will result in a new strain, okay? In a new strain. And this new strain are only partially attacked by our immune system, which therefore results in a milder disease in adults who have previously acquired antibodies. So this is happening in somebody who has had an antibodies to the influenza virus, but they develop mild symptoms. You know how you still get sick a little bit? Sometimes you get the flu shot, and then let's say a month after or 2 months after, you feel like a little bit sick. This is the reason why, because of antigenic drift. Okay. Now, remember this. This antigenic drift is usually not serious and it's mild and it can be self-limiting and most people resolve without any complication. But you have to know this. This is actually serious in older patients. In the geriatric population, it can cause pneumonia and it could be very, very serious. Okay. Now, now we all know about the pandemic. We probably have heard of the pandemic of influenza of 1918. Okay, of 1918. Okay, and basically over a few weeks in 1918, about a 500,000 people in the U.S. Basically died. 12.5 million in India died from it. That pandemic of 1918 was caused by antigenic shift. Here, what happens is that there is a complete change in the glycoprotein spikes, a complete change. Okay, how does this happen? This can only occur with influenza type A. Okay, and the reason is because the mechanism involves trading of RNA segments between animal and human strains. Remember how we said it's segmented? So what happens is that you have two influenza types that co-infect the same cell. Two influenza types co-infect the same cell. They both, once they have taken over that host cell, they undergo replication and they make a new capsid, and RNA segments are embedded in the capsid, and they do a genetic reassortment. This leads to the formation of another virus. This new virus now has a whole new type of glycoproteins, which the human immune system has never, ever been exposed to it before. This means that now the whole population, which has never been exposed to this new strain of glycoproteins, is susceptible to it, which means now everybody can get infected, leading to a pandemic. So the point that I'm trying to make here is that a pandemic happens whenever a virus forms new spikes, new glycoprotein spikes that nobody in the population has been exposed to it. And these glycoprotein spikes are able to attach to our host, to our cells. Okay? So, the pandemic of 1889 was caused by a virus with an H2 hemagglutinin. The 1900 pandemic was caused by a new virus with H3 hemagglutinin. 1918 of the swine flu virus. This is when a swine flu virus was transferred, when it transferred its hemagglutinin to a human virus. So that's why it was called the human swine flu virus. Okay. So that is that about the influenza. Any questions about influenza? This is very important to understanding how the current pandemic is happening. Any questions? Okay. Coronavirus respiratory syndromes. Now, severe acute respiratory syndrome, also called SARS. This is a disease. Another one's called Middle East Respiratory Syndrome, MERS. Both of these are respiratory diseases caused by the coronavirus, which destroy lung cells and can spread via the blood to the heart and the kidneys. These respiratory syndromes of the coronavirus can cause high fever, dyspnea, which is difficulty breathing, body aches, and it can actually progress to pneumonia and dry cough. That's why it can be serious. Okay. In 2003, a lot of people worldwide, about 700, died from the SARS. Okay, we got lucky. Because it was brought under control. The mistake that we did at that time is that the public health people did not make a drug or a vaccine against this one. It could have actually helped us in a way right now. Okay. Fatality rate means that how many people died from it. Of SARS was about 10%. Of MERS, it was about 55%. Okay. I want you guys to know that camels are actually reservoirs of the MERS. The MERS is actually the coronavirus that happens in the desert, which is very hot. That's why coronavirus, hot temperature does not affect it. It does not kill it. UV light kills it, but not hot temperature. Okay. So classification of the coronavirus, COV. This is basically how we write coronavirus. Coronavirus is enveloped linear positive single-stranded RNA virus that has a helical capsid. This coronavirus, what does it cause? Remember, we said it causes common cold. It also causes SARS and it causes MERS. So how do we write these? SARS, coronavirus, MERS, coronavirus. How are they transmitted? Respiratory secretions. Okay. MERS coronavirus is common in the Arabian Peninsula. The SARS coronavirus was common in Asia. It affects all demographics of all ages.